We wanted mammoths. But what if the same tools that could help bring them back could also build something far worse? De-extinction and synthetic biology sound exciting. We talk about reviving lost traits, saving endangered animals, even designing new species to fix broken ecosystems. All of that uses powerful tools, gene editing, DNA synthesis, and now AI that can help design new sequences of DNA and proteins. Used well, these tools could help life. Used badly, they could threaten it. Here's the uncomfortable truth. The tools that can create life can also help create things that harm it. The same systems that could help build a cold-resistant elephant or an engineered coral can, in theory, help design stronger viruses or new toxins. Nobody set out to do that for real in de-extinction projects, but the door is there, and people are starting to worry about who might want to walk through it. Right now, if you want to order custom DNA from most big companies, your order gets checked. They scan the sequence you send against lists of dangerous genes, like known toxins or parts of nasty viruses. If your sequence looks suspicious, they can block the order or ask more questions. This DNA firewall is one of the main things that stands between powerful lab tools and people with bad ideas. But recently, researchers have shown that AI can sometimes design biological sequences that slip past these basic checks. Instead of copying known bad genes, models can generate new ones that behave similarly, but don't look like anything on the blacklist. From a computer's point of view, it's just creative protein design. From a safety point of view, that's a nightmare. It means you could, in theory, create something harmful that doesn't ring any alarms in existing screening systems. For de-extinction and synthetic biology, this is a big problem. Why? Because these fields depend on pushing DNA technology forward. Better gene editing, faster DNA printing, smarter design tools. The more advanced everything gets, the more useful it is for saving species, building climate tools, and studying evolution. But at the same time, those upgrades can make it easier to create things we absolutely do not want in the world. So now we're stuck in a tension. On one side, we have amazing goals: bring back lost genetic diversity, protect endangered animals, maybe even revive parts of extinct species to help damaged ecosystems. On the other side, we have a growing fear: are we also building the perfect toolkit for someone who wants to design a custom plague or a powerful new toxin? That's where biosecurity comes in. Biosecurity is basically safety for biology. It asks questions like, who can access DNA synthesis services? How do we check what people are ordering? Should some AI tools for designing biological sequences be restricted or only used in secure labs? And how do we balance open science, which helps good people do important work, with guardrails that keep bad actors out? For gene synthesis companies, this means they can't just rely on simple blacklist filters anymore. They'll need smarter screening that can spot unknown but suspicious sequences, not only exact matches to known bad genes. They may need human review for risky orders, stronger rules about who they sell to, and better sharing of warning lists across companies. All of this adds time and cost, but without it, the risk grows. For de-extinction projects, it means being honest about the dual-use nature of their technologies. If you're building better tools for editing genomes, you should also be working with biosecurity experts and regulators. If you're using AI to help design traits, you need to think about how those models might be misused if they leak out into the wild. Saying we're just doing conservation isn't enough when the tools themselves are so powerful. The scary version of this story is obvious. Someone takes de-extinction and synthetic biology tools and uses them to make something harmful on purpose. But there's also a softer, slower danger: accidents. Lab error. A sequence that behaves differently than expected. A release that was probably safe based on models until it wasn't. Nature is complex, and our track record of controlling complex things perfectly is not great. But this doesn't mean we have to shut everything down. The question isn't tech or no tech, it's how do we make the tech safer while it grows. That includes better screening, strong international rules, and something we don't talk about enough: humility. We need to be clear about what we don't know, and careful about moving fast in a system as tangled and sensitive as life on Earth. There is a version of the future where de-extinction tools help rescue species, synthetic biology helps clean pollution, and AI designs vaccines faster than any human team could. There is also a version where the same ingredients are used to build things we can't easily stop. Which one we get depends on what rules we put in place now and how seriously we take biosecurity today, not after something goes wrong. On the De-Extinction Lab, we love exploring big ideas about bringing life back and designing the future of nature. But we also have to ask the uncomfortable questions. What else are we unlocking when we unlock these tools? Are we building a safer world for life, or just opening more doors and hoping nobody walks through the wrong one? If you want to keep exploring that sharp edge between hope and risk, make sure to like this video, subscribe to the De-Extinction Lab, and turn on the bell. Because the same tools that might help revive the past could also shape the most dangerous parts of our future.